Hi, I'm Nathan Scalf, and I'm from Lexington, Kentucky.
Hello, I'm Jeremy Liew, and I'm from Carlsbad, California.
Hi, I'm Gehlhoff. I'm from the Cincinnati area in northern Kentucky.
Hi, I'm Jahanna Donegan-Wurz, and I'm from Chittenango, New York.
Hi, I'm Brynn Edwards, and I'm from Fort Lauderdale, Florida.
Hi, my name is Natali Long, and I'm from Richmond, Virginia.
Hello, my name is Nathan Heath, and I'm from Malvern, Arkansas.
Hello, my name is George Taylor, and I'm from Charlotte, North Carolina. Our mentorship consists of Zoe Zlatic, Becky Fritchie, Laura Tomlin, Dr. George Pantelos, and Ms. Phyllis Friola. They help by giving us advice and guidance throughout this entire project.
After designing the mission patch, I'll explain its symbolism. The octagon displays 8 sides to demonstrate the 8 members. Earth demonstrates how this topic will relate back to life on Earth and hopefully improve people's lives. The blood cells in orbit demonstrate how the research focuses on the characteristics of RBCs in microgravity. The NASA worm font, displayed on all the text, represents our ties back to NASA. The star pattern on the background is from the NASA Meatball logo and also shows our connection to this organization. Lastly, the light blue ring also demonstrates our connection to Earth as it displays the colors of our ocean while complementing the patch and making it look more balanced.
This investigation will examine current anemia treatments, analyze their applicability for space hemolysis, and discuss how these treatments can be adapted to better suit spaceflight. Even though this is a new topic of research, there's a multitude of reasons for the cause of space hemolysis, including fluid shifts and more. We started looking into anemia treatments to treat space hemolysis because anemia and space hemolysis are so close, the treatments could potentially be interchangeable. So within our research, we will look into ways that anemia is cured and see if it's applicable to space hemolysis. The goal of this research is to find out how to further prevent health complications with blood throughout long space journeys. Our goal is to be able to see if anemia treatments are applicable to space hemolysis induced in microgravity. When exposed to a period in microgravity, it has been found that astronauts will experience a phenomenon called space hemolysis. During this process of hemolysis, astronauts will experience up to 45% of an increase in the destruction and the rupture of red blood cells or RBCs. The effect of hemolysis continues even after astronauts come back to the Earth setting. We're currently trying to solve this issue because any sort of anemia or anemia-like abnormality will not allow our body to get the sufficient amount of oxygen we need. It can start to damage our vital organs that are reliant on blood, like the heart and brain. Also, the body's tissues will also be starved of oxygen as well. This leads to hypoxia, which shuts down the necessary bodily functions.
So what is hemolysis? Hemolysis is when the destruction of red blood cells, or RBCs, exceeds the rate in which those RBCs are being replenished by bone marrow. Bone marrow being a spongy tissue which is found inside certain bones that produces red blood cells, among other things. Now, how do the bodies of astronauts respond to microgravity? Microgravity being one of the key factors as to why astronauts obtain this adaptation. In short, microgravity is the state in which objects appear to be in a constant state of free fall, because they are orbiting around Earth in a continuous rate. Bodies react to microgravity with an abnormal increase in hemoglobin degradation. Hemoglobin is a protein found in RBCs that carries oxygen from the lungs to the rest of the body and returns carbon dioxide to the lungs, or to the tissues back to the lungs. This is an essential function for transporting oxygen to the cells and allowing for cell respiration. Then there's hematocrit, which is the volume of blood that is occupied by healthy RBCs. When there's hematocrit degradation, it means the blood's oxygen carrying capacity has been reduced. Reducing plasma volume refers to the liquid component of the blood that contributes to 55% of the blood's total volume. When this is reduced, it jeopardizes the circulatory system. Lastly, reducing erythrocyte mass indicates a decrease in the total number of red blood cells. Overall, these bodily responses to microgravity impair the oxygen carrying capacity of the blood. Moving into how space anemia affects the body. For one, there is limited oxygen transportation. This is the main source for the other symptoms, those being fatigue or lack of energy, dizziness, potentially headaches, decreased strength and feeling weaker, abnormal heart functions, which can be heart palpitations, disrupted cognitive ability, so the processes of thinking, observing, and solving are more difficult, shortness of breath, and many more. So what do all these physiological responses lead to? Astronauts' anemia. This is also synonymously referred to as space anemia and microgravity-induced anemia. When hemolysis exceeds a rate of 54%, we know there's a significant imbalance. Reasons for astronauts experiencing this adaptation include factors like radiation exposure, fluid distribution, and microgravity, which our main focus for the project will be on microgravity. Radiation exposure is emission of energy as electromagnetic waves, and it damages RBCs. On the other hand, fluid distribution is the movement and redistribution of bodily fluids due to the absence of gravity, which alters blood volume. Another factor is extended missions with prolonged periods of time in microgravity, which continues to add to this problem's severity.
Measuring and monitoring space hemolysis. In order to properly examine the effects of anemia treatments in astronauts, there needs to be a way to test for anemia. Right now, the most common way to test for anemia is using blood draws. However, for spaceflight, there's an increased risk of complications with this technique, and it requires a large amount of materials to be carried on missions, such as sterilized needles, needle disposal, syringe, blood storage, and testing equipment. An alternative method to this is using a deep learning AI. A study was conducted with this where it analyzed retinal fundus images from the UK Biobank, composed over 114,205 fundus images. It found that this deep learning model was able to detect anemia and quantify blood hemoglobin levels with a 95% confidence rate. Fundus photography is something that's already aboard many space missions, including the ISS, and it can be done a lot more frequently than blood draws.
Possible causes for hemolysis. Microgravity is the most likely option given for these rates of increase are unique to spaceflight. Next, fluid shift is possible due to the body purging off 10% of its fluid volume while under the effects of microgravity. Next, other theories being discussed include radiation, isolation, and confinement. Space hemolysis is a catastrophe. Space hemolysis proves itself to be a major difficulty for astronauts due to its similar effects on the body to anemia. While currently unsolved, looking to treatments for anemia could aid in helping cure space anemia. Space hemolysis does not only affect astronauts while under the influence of microgravity, but effects can extend for around a year after astronauts return to Earth, depending on how long they spent under the effects of microgravity. Severe effects include heart failure, loss in control of red blood cell count, which can lead to unwanted structural changes in organ structure, and a loss of consciousness.
Current solutions have been discovered by space organizations to combat hemolysis, which is when the body destroys red blood cells for the duration someone is in space. For example, astronauts who exercise the upper body will face changes in their fluid dynamics. Not only do astronauts need an improved fitness plan to compensate for microgravity, they will need to enhance their nutrition and vitamin intake. It will also help to understand how the bloodstream works in microgravity. Additionally, pharmaceutical treatments are being tested to see if they can undergo the conditions in space to accommodate the astronauts' health for their neurological and blood systems. Although these results for these treatments aren't guaranteed, scientists at NASA and beyond have numerous studies supporting this research.
Alright, so this image below is one of the two Z-cubes developed by Bob Twiggs. This version is a 10 by 10 by 20 centimeter rectangle, equipped with a 3D printed exterior shell, a data logger module, a mini testing container, and a mini camera. The cube also has multiple other sensors that they can measure, as it's equipped with gyroscopes and accelerometers, and much more. Another interesting additional fact is that the Z-cube was actually inspired by a Beanie Baby box, which Mr. Twiggs found when shopping. In our Z-cube, we have 3 tubes of liquid: blood, water, and urine. The blood and urine are solutions. The blood we made with a mixture of glycerin and water, and the urine out of salt and water. We first reached out to Dr. Pantelos for a blood solution. Once we researched the rest of the liquids, we syringed the solutions into separately labeled tubes. On our final prototype, the different solutions were dyed to ensure that we can identify each of them differently in space. Our main goal is to use the camera feature of the Z-cube to measure the visual changes in the liquids. Specifically, we want to notice the viscosity, which is the fluid's resistance to flow. This information is critical to know how these human bodily fluids may perform in microgravity. For example, it will identify how these liquids perform when attempting something, such as a surgical procedure. Now, we expect all the liquids to stick to the walls of our testing tubes. This is due to the adhesion of the liquids. Considering the viscosity, we also believe that blood will adhere more to the container than the water. Urine and water are expected to perform around the same, as not only is our solution mainly water, but so is the actual product.
Erythropoietin-stimulating agents, or ESAs, are a glycoprotein hormone that stimulates red blood cell production. ESA is a pharmacological reproduction of EPO, created using recombinant DNA and manufactured using transfected Chinese hamster ovary cells, or CHOs. Currently, ESAs are approved for conditions in which RBC production is impaired. However, after some analysis, we concluded that ESA is likely to be an ineffective treatment for space hemolysis. This is because in space hemolysis, the immature red blood cells, or reticulocytes, as well as the mature ones, lack EPO receptors and cease to respond to EPO completely. This study defines space hemolysis as EPO independent. Therefore, a hormone replicating EPO is likely to be unable to stimulate red blood cell production.
So refrigerated blood in the medical world stored for blood transfusions can only be used for 42 days before basically expiring. The minute blood leaves the body, nitric oxide levels drop and continue to drop until the donation is over. After those 42 days, the gas is nearly non-existent and would not be able to deliver oxygen properly. When looking at this huge task of bringing blood to space for the off chance a blood transfusion is necessary, or even just revolutionizing storing blood on Earth, dehydrated blood offers the perfect solution. Dehydrated blood is exactly as it sounds, blood that has a liquid component or has its liquid component removed and transforms into a dry pink powder that can potentially be rehydrated. To rehydrate this blood, sterile water must be added and mixed around. In space, there's a specific bag that would be necessary from the presence of microgravity because there's no hydrostatic pressure. This dual blood bag has the powder on one side and the sterile water on the other, so when it's time to combine the two, the notch in the middle is removed. Then to actually activate the solution, a pump is necessary as well as some manual mixing from outside of the bag. This process has been proved to be successful in small quantities, so the next challenge is moving forward and seeing if larger volumes can be taken or be sufficient for the extent of the space travel. Rehydrated blood is thought to mend oxidative damage from microgravity by replenishing the nitric oxide levels and ensuring proper oxygen delivery to tissue. This solution addresses the challenge of storing and using blood in space, potentially providing a reliable way to manage anemia during extended missions.
The preliminary schedule consisted of first coming up with ideas regarding the experiment, and then receiving the Z-cube and creating ideas to apply the subject. And after creating these ideas and applying them, we altered the project to see if there were any flaws. And lastly, we did more research and created the slides.
Possible causes of hemolysis in spaceflight include radiation, fluid shifts, and oxygen needs lowered. Treatments for this condition include surgery, blood transfusion, and supplements. In conclusion, we can see that hemolysis is one of the many problems we face as we venture more into space. This debilitating condition breaks down the red blood cells of astronauts at an increased rate of 54% in space compared to here on Earth. This causes anemia, which starves the vital organs of oxygen, leading to death if gone untreated. This is why it is critical for us to find a suitable treatment methods to use on space flights to assist the astronauts with this condition on long space journeys.
Our team has worked tirelessly on figuring out how hemolysis would react in microgravity. Our goal for this mission is to examine anemia treatments by studying hemolysis in microgravity. Our mission aligns with NASA's strategic goal of expanding human knowledge, capability, and opportunity in space. Future research could lead to discovering a treatment to alleviate hemolysis. Furthermore, learning about how fluid dynamics behave in the upper body will support the data behind why astronauts have swollen faces in space.
My name is Nathan Scalf, and I would like to thank the SEAS program and staff for this fantastic opportunity. Being from Kentucky and having this passion for all things related to spaceflight and science, this program has given me an opportunity to experience the things that I love and wouldn't have been able to get from where I am, while giving me the chance to do real research and meet many influential people living the dream that I wish to have.
I would like to thank the entire SEAS program for this experience, especially to the teachers and coordinators that kept us in order. It was fun to talk to the professors about life in microgravity. Each person offered me a unique perspective and take on space. The dorming experience was also pretty cool. It was my first time doing something of the sort, and I had a really good time. All in all, this program has given me connections with all sorts of people, with all sorts of different expertises, memories of all kinds, and is something in general that I will not be forgetting soon. I highly recommend this program, and I give it a 10 out of 10.
This internship has truly been an incredible experience. I've gotten to meet people from across the country and share a common interest with all of them. It has been wonderful to spend the past week surrounded by people who all have the same love and interest for space exploration as I do. Additionally, I have learned so much from every speaker and mentor at this program. Being able to connect with real professionals in the field has been amazing, and I truly have learned so much in just 1 week. I'd also like to give a special thanks to our group's mentors, Zoe Zlatic and Becky Fricci.
I've had such a wonderful time at this internship. I've learned so much about our subject of research. This has been a wonderful experience to connect with adults in the science industry and the students that have the same interests as me. I've been able to meet people with a variety of backgrounds and cultures, and it's been truly amazing to interact and share ideas. I owe a special thanks to all of the people that have helped guide us, like our mentors and our teammates. I've truly met some really remarkable people. I'm done.
Hi everyone, I would like to give a big thank you to the SEAS program and all the people who worked to make it happen. I've had such an amazing experience all the way from learning something new and fascinating each day to becoming great friends with my whole team. It has been so rewarding to participate in this program, and I know that in years to come I will look back fondly on this time. I think it was incredible. I was able to not only discover more about my passions, but learn some lessons that apply to many aspects of life. I hope you were able to learn something new from this presentation too, just like I have. Thanks again.
I've had the wonderful opportunity to fly down to Austin, Texas from Richmond, Virginia this week to learn about space exploration and microgravity. I have been so fortunate to work with 7 talented, bright individuals from across the state, along with fabulous mentors who have provided guidance and support throughout this entire journey. Together, we decided to study a pressing issue regarding human health and microgravity and spent hours researching, understanding, and learning about hemolysis in space and what treatments will support anemic patients on Earth.
My name is Nathan Thomas Heath, and I've been flown out from Malvern, Arkansas. I'd like to thank the SEAS program for the opportunity it's granted me to pursue my interest further in STEM and for the opportunities of my teammates who have been a very great help in this program. I'd like to thank everyone for their part played and for the interest that this program has been able to grant me further, and it's encouraged me to pursue my interest in the STEM field and a career in it.
Flying all the way from Charlotte, North Carolina to Austin, Texas for this internship has been such an enlightening experience for me. During this time, I have had the wonderful opportunity to meet and connect with new people of different cultures, learn how much research goes into the development of new ideas and in turn, products, and last but not least, I've been granted the ability to stay in contact with many important and helpful people. I believe I can speak for all of us when I say that this internship has changed our lives for the better. With the knowledge, skills, and friends that we've made during the NASA STEM Enhancement in Earth Science program, we wholeheartedly believe that this experience has been a catalyst in the progression of our generation. Thank you for your time.